For centuries, we've been told a story that has shaped the way we see ourselves, our future, and our very existence. This story says that your genes are your destiny, that the moment you were conceived, your future was written in code, a sequence of A's, T's, C's, and G's that you could never escape. But what if I told you that this story is a lie? What if the true power to shape our lives, health, and futures doesn't lie in genetic code alone, but in something intangible, beliefs? Prepare to explore the intersection of mind and matter, a dimension where perception shapes biology and beliefs sculpt reality. This is the science they never taught you. Be sure to stay until the end as we uncover the secrets to controlling your genetic destiny and literally bend your reality. Get ready, humans. Welcome to Astral Breeds, True Oneness. Be sure to hit that like and subscribe button before we begin and join the Astral Breeds Collective. Now, fellow humans, let us start. Since the discovery of DNA in 1953 by James Watson and Francis Crick, modern science has framed this molecule as the blueprint of life, a code that determines who we are, from the color of our eyes to the diseases we might suffer from. Genetic determinism, as it's known, states that we are prisoners of our biology, bound by a molecular script written long before we were born. From cancer to heart disease, from personality traits to intelligence, the message has been clear. Your genes are your fate. But there's a problem with this narrative. It's incomplete. In fact, it's dangerously misleading. We've all heard it before. It runs in the family. Whether it's cancer, diabetes, or heart disease, we've been conditioned to believe that our genetic inheritance is an unavoidable fate. The Human Genome Project, completed in 2003, was supposed to be the key to unlocking the mysteries of these inherited conditions. Yet, what scientists found was far from what they expected. When the Human Genome Project mapped our DNA, scientists expected to find around 100,000 genes, one for each of the proteins that make up the human body. Instead, they found just over 20,000. Worse, they realized that less than 2% of these genes are actually responsible for encoding proteins. So what was the rest of the genome doing? Instead of finding a clear genetic roadmap, scientists discovered that genes are more like potential instructions that require input to be activated. And this is where the story gets interesting. The remaining 98%, once dismissed as junk DNA, has emerged as anything but useless, believed today to not contain instructions for encoding proteins, essentially serving no purpose. However, breakthroughs in molecular biology have challenged this assumption. Dr. John Maddock, a pioneer in genomics, has noted that much of this non-coding DNA is involved in a hidden regulatory network that controls gene expression, called the epigenetic regulatory network, a hidden layer that determines when, where, and how genes express themselves. What Dr. Maddock highlights here is a sophisticated layer of genetic control embedded within the so-called non-coding or junk DNA. This hidden regulatory network involves sequences that act as switches, timing mechanisms, and spatial guides for gene expression, subtly determining which genes are active or inactive in different cells at various stages of development and under specific conditions. Maddock's insights suggest that non-coding DNA is central to how complex life organizes itself and responds to its environment. Effectively, the ERN interacts dynamically with environmental factors, modulating our gene expression and contributing to the unique individuality of each organism. This vast, unseen architecture appears to orchestrate complex processes like development, aging, and even aspects of consciousness. Recent breakthroughs led by projects like the ENCODE Project Consortium suggest that non-coding DNA manipulates biological activity, acting as switches and regulators for gene expression. As Dr. Ewan Birney, ENCODE lead scientist, explains, these so-called junk regions apparently help fine-tune gene expression, allowing for the complex orchestra of cellular functions that sustain life. Two primary mechanisms that regulate gene expression are DNA methylation and histone modification. DNA methylation involves the addition of methyl groups to specific DNA regions, often silencing genes by preventing transcription factors from binding. Histone modification, on the other hand, alters the structure of chromatin, the protein DNA complex, by adding or removing chemical groups to histone proteins around which DNA is coiled. This process can tighten or loosen chromatin, making genes more or less accessible for transcriptional machinery, thereby regulating gene activation with precision. Non-coding DNA appears to influence which genes are active in different tissues, shaping organ development and the immune system's ability to respond to pathogens. Studies by Dr. David Hausler at the University of California, Santa Cruz suggest that variations within non-coding regions may even contribute to uniquely human cognitive abilities, linking it to higher-level functions like reasoning and language. Epigenetics, an advanced field at the intersection of molecular biology and environmental science, has revolutionized our understanding of gene expression by revealing that DNA is not a static genetic code, but a dynamic system finely attuned to external influences. Contemporary research now shows that factors such as diet, stress, exposure to environmental toxins, and even psychological states can trigger epigenetic modifications, which adjust gene expression without changing the underlying DNA sequence. Dr. Bruce Lipton's pioneering work highlights how these modifications function as precise molecular switches, enabling genes to activate or deactivate in response to external conditions. This process also underscores a crucial, previously overlooked role for non-coding or junk DNA, positioning it as an active participant in an ongoing interaction between our genetic code and environmental factors. This means that, in a very real sense, our lifestyles, habits, and environments can alter our biological destiny. In terms of aging, non-coding DNA may play a critical role. Dr. David Sinclair, a geneticist at Harvard University, has discovered that specific RNA molecules from these non-coding regions are involved in the aging process by influencing DNA repair. Such findings are crucial as they may pave the way for anti-aging therapies and age-related disease interventions. Neurodegenerative conditions, including Alzheimer's, have been linked to the malfunction of non-coding RNA molecules, suggesting that these areas of DNA might be key to treating or even preventing such disorders. Recent studies demonstrate that epigenetic changes can even be passed down across generations, a phenomenon known as transgenerational epigenetic inheritance. This breakthrough implies that our ancestors' life experiences, whether traumatic events, dietary patterns, or environmental exposures, can leave a lasting impression on our own genetic makeup, influencing how susceptible we are to various conditions. This adaptability is part of how our epigenome integrates external and internal cues, creating a flexible yet highly regulated gene expression landscape. The traditional belief that genes control our biological fate is increasingly challenged by this new science. The idea that we are programmed by our DNA to have specific traits or illnesses is losing ground as we realize how much influence we hold over genetic expression. There's a growing body of evidence suggesting that genetic information can be influenced by what some call a biological internet. Research has shown that DNA emits low-level electromagnetic fields and can even communicate with other DNA molecules at a distance, transferring information without any physical or chemical contact. This concept of DNA as a bioelectromagnetic transmitter is controversial, yet studies in cellular resonance imply that cells and genes may interact in ways that challenge traditional biological models, hinting at an interconnected field of life where our genes are but one node in a vast network of biological information. Cutting-edge studies now suggest that our genetic material may not only respond to physical and emotional cues, but also to biofields, subtle electromagnetic fields generated by our bodies that extend beyond the limits of our skin. The human biofield, often referred to as a type of energy field, is gaining attention in scientific communities as researchers explore how electromagnetic signals in and around the body might influence gene expression. This biofield may be a critical missing link that mediates how emotions, beliefs, and even thoughts directly influence our physical health at the genetic level. The biofield theory posits that every cell emits an electromagnetic signature, creating a dynamic energy network that carries information across the body. Preliminary studies suggest that the biofield might interact with gene expression by altering the electromagnetic properties of the cellular environment. For example, researchers have found that certain frequencies can stimulate or suppress specific genetic sequences, hinting at a previously unknown layer of genetic regulation. This suggests that the biofield may act as a conductor, orchestrating the music of gene expression in ways we are only beginning to understand. This field is also probing a phenomenon called remote genetic influence, where it appears that cells exposed to specific electromagnetic frequencies can affect the gene expression of distant cells without direct contact. Although still experimental, early data points to a radical possibility that we might one day influence gene expression in parts of our bodies through targeted frequencies or even our own biofield vibrations. Imagine harnessing specific brainwave patterns, such as those generated during meditation or peak emotional states, to activate healing genes in organs or tissues elsewhere in the body. Studies in biofield science have shown that a state of mental coherence where mind, heart, and emotions are harmoniously aligned produces measurable changes in our biofield, which can impact gene expression in remarkable ways. Practices like meditation, deep breathing, and heart-centered techniques have been found to enhance this biofield coherence, aligning it with cellular processes and promoting a state of resonance that seems to support cellular repair and optimal gene function. This theory suggests that we might be able to consciously tune our biofield, thereby actively guiding our genetic blueprint toward well-being. This emerging view adds yet another layer of agency and empowerment in the gene-environment dynamic, positioning us not only as active participants in our genetic expression, but as conscious co-creators in our evolutionary journey. If future studies confirm the potential of biofield interaction with genetic material, the implications could be profound. This would mean that by fine-tuning our internal and external energetic environments, we might transcend current biological limitations, enhancing everything from disease resistance to lifespan by simply tuning into our own vibrational states. Imagine a future where we hold the reins to our own biological evolution, not through invasive technology or genetic engineering, but by tapping into the power of our biofield, the energetic blueprint that connects mind, body, and soul. What do you think, fellow human? Could this really be the future of human evolution, where we transcend our limits simply by tuning into our own vibrational frequencies? Be sure to share your thoughts in the comments. We'd love to hear what you think. So now, as we peel back the layers of genes, a profound truth begins to emerge from the shadows, that what we believe about ourselves may be just as powerful, perhaps even more so, than what science has long assured us we are. What if DNA, the sacred code of life, isn't an unyielding script locked in place, but a fluid language, shifting and bending in response to our deepest perceptions? What if, within this so-called genetic blueprint, lies a dynamic dialogue, a conversation between who we think we are and who we could yet become? What do you think? Thank you, humans. If you enjoyed this video's exploration into the mysteries of genes, please leave us a like and comment a powerful thought below. If you wish to know more secrets of the forces shaping your reality, be sure to hit that subscribe button and join the Astral Breeds Collective for more enlightened journeys. Don't forget to ring that notification bell and share it with your fellow Earthling. Until next time, fellow humans, and control your mind.